Hello friends, welcome back to a new video that is MCQs on cell signaling, and this is the first part of this series of videos on cell signaling. Okay, so the first question of this video, that is, which of the following is not a commonly observed consequence of binding of a signaling molecule to its cell surface receptor? So what is not common with the binding of a signaling molecule to its cell surface receptors? So options are option A, that is receptor dimerization, option B receptor phosphorylation, option C conformational changes in the receptor, or option D increased synthesis of the receptor. So which one is not common with the binding of signaling molecule to its surface receptor? So the right answer is option D, that is increased synthesis of the receptor. So this is the wrong option, but the other three, that is the receptor dimerization, receptor phosphorylation and conformational changes in the receptor, can be done by this binding of the signaling molecule to its cell surface receptor. Okay, so this is the first one. So next move on to question number 2, that is the mechanism of signal transduction by steroid hormones differs from amine and peptide hormones because option A, steroids use small water soluble second messenger, or option B, they bind with specific receptor proteins on target cell plasma membrane, option C, they bind to cytoplasmic or nuclear receptors and affect gene expression, or option D, they are secreted from exocrine glands. So which one is the right answer about the signals transduction by steroid hormones, which is differ from amine and peptide hormones? Because option C, option C is the right answer. So option C is they bind, that means they bind to cytoplasmic or nuclear receptors and affect gene expression. This steroid hormones affects the gene expression. That's why they differ from amine and peptide hormones. Okay, so let's move on to question number 3. That is, choose the following mechanism by which cell communicate with each other. By which mechanism cell communicate with each other? So there are the options. Option 1 is transcription factors are secreted from one cell and taken up by the target cell where they influence gene expression. This is the first one. Next is cell surface molecules on adjacent cells interact and initiate a signal transduction process that influence cellular behavior and gene expression. Option 3, cells from gap junctions with adjacent cells allowing the passage of small molecules. Option 4, that is cells secrete proteins and small hydrophilic molecules which interact with cell surface receptors on target cell and initiate a signal transduction process that influences cellular behavior and gene expression. So which one? So here the options, which arrangement of option is the correct one? So option A is 1 and 2 statement is the correct. Option B 2 and 4 is correct. Option C 2, 3 and 4 is correct, or option D 1, 2, 3, 4, all options are correct. So which one is the correct option? So correct is option C, that is 2, 3, 4 is the right statement about the cell communication. 2, 3 and 4 is the right statement. The first one is not the right statement about cells communication. So transcription factors are secreted from one cell and taken up by the target cell where they influence gene expression. So transcription factor not secreted from one cell and taken up by the target cell where they influence the gene expression. This is the wrong statement about cell communication. Okay, so let's move on to question number 4. That is, protein phosphatase, or option A, catalyze the addition of phosphate residues to proteins. Option B, catalyze the removal of phosphate residues from proteins. Option C, catalyze the addition of glycophosphatidylcholine inositol to proteins, or option D, these protein phosphatase are proteins that specifically bind phosphorylated proteins. So which one is the right answer about the protein phosphatase? So the right answer is option B, that is this protein phosphatase catalyze the removal of phosphate residue from protein. That is the role of protein phosphatase, to remove the phosphate group from protein. Okay, so let's move on to question number 5. That is, intracellular receptors are option A, as usually bind hydrophobic ligands. Option B, may be located either in the cytosol or nucleus in unbound state, or option C, when bound to their ligand regulate gene transcription, or option D, that is all of the above are correct ones. So which is the right answer about intracellular receptors? So the intracellular receptors are option D is the correct one. That is, all of these statements are correct about intracellular receptors. That means they are as usually bind hydrophobic ligands. They may be located either in the cytosol or nucleus in unbound state, or they can when they bound to their ligand regulate gene transcription. These all statements are correct about intracellular receptors. Okay, so let's move on to question number 6. It is, which of the following conditions promote tumor growth? And the options are option A, increased expression of MHC, option B, increased expression of Th2 cytokines, that is T helper cell 2 cytokines, or option C, increased expression of co-stimulatory molecules, or option D, decreased expression of CTLA4. So which one is correct answer about the tumor growth promoting condition? So the right answer is option B, that is increased expression of Th2 cytokines. So which of the following condition promote tumor growth? That is increased expression of Th2 cytokines. Okay, so let's move on to question number 7. That is, retrograde signaling involves communication of option A, nucleus to the chloroplast, option B, endoplasmic reticulum to the nucleus, option C, nucleus to the mitochondria, or option D, chloroplast to the nucleus. So which one is the right answer about the retrograde signaling? So the right answer is option D, that is chloroplast to the nucleus. So retrograde signaling involves communication of chloroplast to the nucleus. So let's see what is retrograde signaling. Retrograde signaling is a process where a signal travel backward from a target source to its original source. So this type of signaling typically occurs between the mitochondria or chloroplast to their nucleus. Okay, so this is called the backward signaling. So normal signaling is from nucleus to chloroplast or mitochondria or any other organelle, but the backward signaling is from mitochondria or chloroplast or any other organelle to the nucleus. Okay, this is called the retrograde signaling. So let's move on to question number 8. It is cytochrome C, sorry, cytochrome C is normally found in the inner mitochondrial membrane. So it is released into the cytoplasm during option A, apoptosis, option B, necrosis, option C, cell differentiation, or option D, cell proliferation. So which one is the right answer? So the right answer is option A, that is apoptosis. So during apoptosis, cytochrome C released into the cytoplasm. Okay, so this is the right answer, option A. So next question देखेंगे. Next is question number 9. So signal recognition particle consists of option A, a single large RNA, option B, multi-protein complex, option C, a complex of single RNA and multiple proteins, or option D, a complex of multiple RNA and multiple proteins. So which one is correct about the signal recognition particle? And the right answer is option C, that is a complex of single RNA and multiple proteins. So signal recognition particles consist of a complex of single RNA and multiple proteins. Okay, so what is signal recognition particle, SRP? So this SRP is a ribonucleoprotein, that is RNA with protein complex that targets specific proteins to the endoplasmic reticulum in eukaryotes and the plasma membrane in prokaryotes. So it has two functions. First one, this SRP targets proteins with a signal sequence to the endoplasmic reticulum, and the second function is SRP protects mRNA from degradation. Okay, so these are the two roles of this SRP or signal recognition particles. Okay, so let's move on to question number 10. That is which which of the following has the highest affinity of ligand receptor interaction? So the options are option A, insulin receptor, option B, lectin sugar receptor, option C, biotin avidin, or option D, cytokine receptor. So which one has the highest affinity of ligand receptor interaction? So the right answer is option C. Option C, that is biotin avidin interaction is the highest affinity of ligand receptor interaction. So what is avidin? Avidin is a tetrameric glycoprotein and each of its 4 monomer can bind biotin. So avidin binds biotin very tightly and specifically. So this interaction between avidin and biotin is one of the strongest non-covalent interaction in nature. Okay, so let's move on to question number 11. It is molecular basis of Chereck-Higashi syndrome is option A, mutation in a protein involved in regulation of intracellular trafficking, option B, due to deficiency of adenosine deaminase, option C, error in antigen processing, or option D, reversal of energy in self-reactive clones. So which one is correct about molecular basis of Chereck-Higashi syndrome? So the right answer is option A, that is mutation in a protein involved in regulation of intracellular trafficking. So molecular basis of Chereck-Higashi syndrome is mutation in a protein involved in regulation of intracellular trafficking. Okay, so this syndrome is caused by a mutation in the LYST gene, that is LYST means lysosomal trafficking regulator, also known as CHS1. So this syndrome is caused by mutation in the lysosomal trafficking, so trafficking of lysosome. Okay, so giant lysosomal granules develop in neutrophils and other cells. So the abnormal lysosomes cannot fuse with phagosomes. So ingested bacteria cannot be lysed normally. That is the syndrome. That is this Chereck-Higashi syndrome. Okay. So let's move on to question number 12. It is cholera toxin cause massive and often fatal diarrhea by option A, inactivating G1 protein, option B, irreversibly activating adenylate cyclase, option C, locking GS protein into an inactive form, option D, rapidly hydrolyzing G protein GTP to GDP, hydrolyzing G protein from its GTP form to GDP. So which one is correct about the cholera toxin? So the right answer is option B, that is irreversibly activating adenylate cyclase. So cholera toxin causes massive and often fatal diarrhea by irreversibly activating adenylate cyclase. So how it activating adenylate cyclase? So here we will see in this picture. So let's see this. So this is the mechanism of action of cholera toxin. So here you can see this portion is the toxin. This is the toxin, cholera toxin, and when it bind to the ganglioside receptor, this is the receptor. When it binds to the receptor, only the A subunit of this toxin enter into the cell, and when it enter into the cell, it binds with the G protein. It binds with the G protein, and it after binding with G protein, this increases the adenylyte cyclase activity. This binding with G protein with this toxin subunit increases the adenylyte cyclase activity, and this adenylyte cyclase activity increase the CAMP in this cell. So because of this CAMP cell is increased, so it affect the membrane channels, and ultimately the nutrients or minerals come out from the cell to the outside through these channels. Okay, so loss of cell nutrients occur, which is known as diarrhea. Okay, so this is the process of cholera toxin, how they perform diarrhea in our body. Okay, so let's move on to next question. That is question number 13. So, which of the following is not a consequence of increased cellular level of CAMP? So, options are option A, activation of a kinase cascade, option B, activation of the transducing G protein, option C, increased phosphorylation of glycogen phosphorylase, or option D, dissociation of the CAMP dependent protein kinase tetramer. So, which one is correct option? So the right answer is option B, that is activation of the transducing G protein. So which of this following is not a consequence of increased cellular level of CAMP? So activation of the transducing G protein is not the consequence of increased cellular level of CAMP. Okay, so let's move on to question number 14. It is, which one of the following amino acid in protein does not undergo phosphorylation? So which cannot perform phosphorylation? And the options of amino acids are option A, serine, option B, threonine, option C, proline, or option D, tyrosine. So which one cannot perform phosphorylation? So the right answer is option C, that is proline. Proline cannot perform phosphorylation because to perform phosphorylation there have to perform a bonding between phosphate group with the oxygen group. So here no free oxygen group is present in their side chain. Okay, so that's why no phosphorylation can be done. But here you can see in serine, this is a OH group is present in threonine. Here a OH group is present in the side chain, and in tyrosine there is a OH group is present in the side chain. So that's why they can perform phosphorylation with the phosphate group. Okay, so this is the reason proline cannot perform phosphorylation. So next question देखेंगे. Next is question number 15. It is, an extracellular ligand will option A, elicit the same response in various cells that have a receptor for the ligand, option B, elicit the same response but to varying degrees in various cells that have a receptor for the ligand, or option C, may elicit different responses in various cells that have a receptor for the ligand, or option D, elicit the same response in all types of cells because receptors have to be identical to bind to the same ligands. So which one is correct about the extracellular ligand? So the right answer is option C, that is an extracellular ligand will may elicit different response in various cells that have a receptor for the ligand. So extracellular ligand will perform differently in different cell where they have a receptor for the ligand. Okay, so next question देखेंगे. Next is question number 16. That is, all of the following statements about monomeric G protein are true except option A, they are regulated by GTP GDP exchange proteins, option B, they are regulated by GTPase activating proteins, option C, they regulate vehicle fusion, or option D, they help in uncoating of vesicle. So which one is right about this G protein? Which is not right actually. So the right answer is option D, that is they help in uncoating of vehicles. So all of the following statements are monomeric G protein. Statements are all about monomeric G protein except this last option, that is they help in uncoating of vehicle. This is not true. So for this is the right answer, because except this option all these three are about this G protein, monomeric G protein. Okay, next question is question number 17, that is the amino acid sequence of a novel membrane protein contains 4 immunoglobulin like domains and 6 fibronectin like repeats. So this protein is most likely a option A hormone responsive ion channel, option B cell adhesion molecule, option C G protein, option D transcription factor. So which one is right? The right answer is option B, that is cell adhesion molecules. So the amino acid sequence of a membrane protein contain 4 immunoglobulin like domain and 6 fibronectin like repeats. This protein is most likely a cell adhesion molecule. So what is cell adhesion molecule? These adhesion molecules are cell surface protein that mediate the interaction between cells or between cells and the extracellular matrix. Okay. So next is, which of the following types of genetic changes is least likely to be found in an oncogene in a tumor? Option A gene amplification, option B chromosome translocation, option C missense mutation, option D nonsense mutation. So which of the following types of genetic changes is least likely to be found in an oncogene in a tumor? So the right answer is option D, that is nonsense mutation. So nonsense mutation is the least likely to occur in oncogene. So what is nonsense mutation? A nonsense mutation is a change in DNA that causes a protein to end its translation earlier than expected by the stop codon. Okay. So next question देखेंगे. Next is, protein phosphorylation is part of most pathways of intracellular signal transduction. All of the following statements about phosphorylation are correct except option A, phosphorylation can be on a serine or threonine. Option B, phosphorylation changes the structure and function of the protein. Option C, protein kinase cascade result in amplification of the signal. Or option D, that is tyrosine kinases are always a part of a receptor protein. So which one is correct? So the right answer is option D, that is tyrosine kinase are always a part of the receptor protein. So this option, this last option, it is option D, is not the concept, not about this phosphorylation. So protein phosphorylation is part of most pathways of intracellular signal transduction. So all of the following statements, is option A, B and C, is all about phosphorylation, but except option D. This is not the correct. That is tyrosine kinase are always a part of receptor protein. This is not true. Tyrosine kinase is not always part of this receptor protein. Okay. So next, that is the last question of this video. It is CAMP activates CAMP dependent protein kinase by option A stimulating its phosphorylation, option B stimulating the dimerization of kinase subunit, option C increasing its expression by stimulating the release of a translational inhibitory protein bound to its mRNA, or option D binding regulatory subunit and inducing their release from the catalytic subunit. So which one is right about the CAMP activates CAMP dependent protein kinase by option D, that is binding regulatory subunit and inducing their release from the catalytic subunit. So CAMP activates CAMP dependent protein kinase by binding regulatory subunit and inducing their release from the catalytic subunit. Okay, so these are all 20 questions of this part 1 video on cell signaling MCQs. Okay, so thank you for watching this video.